Hello. I'm a medical school student at Gazi University, and as John mentioned, I was his intern at JCVI for this summer. We talk about and possible new phenotypes by using BactoBox. Us, for drinking water safety, clinical diagnostics, and of course for the research purposes. And we do have some really good methods for the enumeration, just like CFU, colony forming unit. CFU can give us the live and dead distinction, but it takes up 1 to 3 days. And normally one bacterium yields one colony, and this is okay. But as you might know, our cell clumps. So you see hundreds of minimal cell here clumped as one. So one clump yields also one colony in CFU, and this is a source of errors. And we also have picogreen assay. It takes up to 1 day to get the results. It is really accurate, but it gives us the total DNA number only. And it cannot also make the live and dead distinction, and it's labor intensive. So we also have one more method that's called CCU, color changing unit. We can make live and dead distinguish pretty well with CCUs, but it takes up to 7 days to get the results, and it gives you a range, not a precise number. And it's labor intensive too, and it's really easy to mess up. The cross-contamination risk between wells is really high, and it looks like this when you contaminate the wells. So we basically make a 10-fold serial dilution, just like this, all the way to the down. And our cells pretty much look like this in the wells. And here's the thing. The media that we use has phenol red in it, which is a pH indicator. And our cells produce acids when they're metabolically active. So when there are cells if there's any cells in those wells, that well just turns to yellow, and this is how we make the enumeration by using the CCU, and that's why we only get the range, as you can see here. So here is a real CCU plate, and here is the machine that's called BactoBox. BactoBox is great because it can give us the results in 5 to 10 minutes. It gives us the precise measurements, and it's really easy to use. It also has some downsides, like no live and dead distinction, and clumping errors still exist. And here I come to hopefully solve those problems and also look for a possible new phenotype. So let me also explain how we do the measurement with BactoBox. We take 101 microliter from our cell culture, put it into 10 mil PBS, and we just vortex it and push the measure button. So I did first the growth curve experiment for JCVI Syn3B with the BactoBox. And for that, I just grow my cells into 100 mil media. And I had more than 20 time points. And at each time point, I took out 1 mil. And I first did the conventional method. It's measurements. So after some good amount of measurements, you see the pipette tip boxes that I work. And it's really easier. And after all those and in this growth curve, this comes from BactoBox, we can observe log state. Here is the graph for JCVI Syn3B. The blue line is from the BactoBox, and the range comes from CCU. As you can see that the first 2 days, it correlates really well. And I also did the same thing for Mycoplasma mycoides, which is, as I told you before, pretty much the same thing with JCVI Syn1. And as you can see, for the first 2 days, it also correlates really well. So to understand better the results and to deep dive to the data that comes from BactoBox, let's get a better understanding about the BactoBox. First of all, BactoBox is an impedance flow cytometer. And let's start with what is impedance. So impedance is basically opposition to the alternating current combined by resistance and reactance. So resistance, we all know from high school, is that opposition to the current flow in both alternating current and direct current. circuits. So, but the reactance only occurs in alternating current, and is opposition to the alternating current caused by capacitors and inductors. So why this is important is because actually cell membrane acts as a biological capacitor. So let's zoom into the cell membrane and compare it with a capacitor. As you can see here, capacitors have two conductive plates, whereas the cell membrane has intra and extracellular fluids. Capacitor have a dielectric layer, where cell membrane has lipid bilayer, which acts like an insulator. So that's why we can say that capacitor acts like a biological capacitor. The cell membrane acts like a biological capacitor. So here you see a cell, and BactoBox has basically the two electrodes, and in those electrodes there is an electric field. And when a bacteria passes through those electrodes, just like this, it interrupts the electric field there. And this is really important to BactoBox because BactoBox can read those changes in the signal and evaluate it as there is a cell. So, and BactoBox can make measurements with two different frequencies. To understand why we do that, we need to understand the relationship between the capacitor reactance and impedance. So let me show you how we calculate the impedance. First, we get the resistance and capacitor reactance, and we calculate the impedance just like we do it for the hypotenuse that by using the Pythagorean theorem. So if we would increase the capacitance, would also, increase. So let me show you the capacitor reactance formula and relationship between the frequency and impedance. So when we make measurement at low frequency, capacitor reactance goes up, and because of that, low frequency current cannot pass through the cells. And since they about the external properties of cells like shape, size, etc. When we make the measurement with high goes down, that's why impedance goes down. So high frequency current can pass through, and it tells more about the internal properties of the cells. And there is one last thing that I need to cover before looking at more data, the phase. Normally, voltage and current are synced together. But when there is a capacitor in the circuit, voltage falls behind, and it looks like this. And this is what we call the phase shift. Phase shift is actually the when the voltage falls behind the current. And this is a thing that BactoBox can also measure. So now I'm going to show you some phase distribution graphs. So I have some measurements from first day to the all the way to the 8th day, and we can observe a very beautiful shift to the left on the phase distribution graph. And we also observe the same thing with high frequency too. And let me simplify this and get 3 time points and invert this graph to line up with our growth curve to see better what's going on. So you can see how the shift, we can see the shift better now. And I also did a heat experiment with the JCVI SYN1 by using the BactoBox. Here I basically get 10 different temperatures, and I set my water bath to that temperatures, and I heated them. I treated them with heat for 15 minutes, and I did CCUs to understand how many of them are alive at that temperature. And CCU tells us that up until the 50 centigrade degrees, they are alive, but after 52, there are no cells alive. So I just get the phase distribution data from the BactoBox for those temperatures too. And we can also see a very beautiful shift here. And when we especially draw a line, as you can see here, we can see that. Now let's remember that SYN1 can survive up to 50 centigrade degrees. And now let's look up the graphs that, phase distribution graphs that up to 50 centigrade degrees. As you can see, after 50, until 50, there are they falls to the right side. But when we look up to the ones who are, more than 50 centigrade degrees, they falls to the left side. So there is a very clear shift on the high frequency. And we can also observe a frequency for the, we can also observe a shift at the low frequency too. Even though it's not obvious, it's still clear shift. So as a conclusion, I did the growth curve analysis for JCVI SYN3B and SYN1 by using BactoBox, and we can say that it really correlates well with our golden. We also observed a new measurable phenotype for JCVI SYN3B and SYN1 that changes as the cell culture. Observed this new measurable phenotype for JCVI SYN1 with the heat experiment too, which is great for the labs that works with our cells, minimal and synthetic cells. This new phenotype. So I do owe a special thanks to Rudy, National Science Foundation, and John Glass for making this experience possible for me, and Zumra, Tyler, Ron, and JCVI. Thank you all very much for listening to me, and I can answer your questions now.
Yavuz, so this instrument, you also, in this process, you use my diabetes glucometer to measure the utilization of glucose during this, and got some nice data with that. And we are trying to, you might talk about other deep dives into the data that we're hoping to get into with the manufacturer of the instrument, SBT Instruments, which is a Danish company.
So I guess more thing that we can do with BactoBox, because I just show you the low and high frequency results. We can also get the amplitude data, and when by combining all these data, we can also measure the opacity of the cell membrane for this electricity and all this kind of stuff. Those are the new features, but we can get this out from the data we have, and I'm sure that it will also tell a different story about our minimal and synthetic cells. And maybe one interesting thing that I would say, that when I do the heat experiment, the numbers were the numbers were interesting. For example, 65 centigrade degrees were pretty much the same with the 37. But when you look at the phase distribution data, you could see that difference really well. So I think when we, play with the data and, combine it together, we can get more understanding about our cells.
Did you also show that the minimal cell was more susceptible to heat than the wild type cell?
Actually, it is almost the same. For 50 centigrade degrees, they seems like there are more alive cells there, but for 52, they both dead.